Welcome to BioSignals, the podcast powered by BioCrossroads, where we decode the pulse of Indiana's life sciences sector. Each episode, we bring you the innovators, breakthroughs, and bold ideas shaping the future of health and science. In this episode of BioSignals, Vince Wong will sit down with Milan Radovich, a nationally recognized leader in cancer genomics and precision oncology. Milan's work sits at the forefront of translating complex genomic data into real-world clinical insights, helping redefine how cancer is diagnosed and treated. Milan's research has been instrumental in advancing biomarker-driven approaches, enabling more personalized and effective patient therapies. In this conversation, we will explore how precision medicine is evolving, the challenges of turning data into action in clinical settings, and how Indiana's unique ecosystem is accelerating the path from discovery to patient impact. We'll also discuss the future of oncology, the role of AI and data science, and what it will take to bring the next generation of therapies to patients faster.
It's my pleasure to have Dr. Milan Radovich, who's the Chief Science Officer at Caris Life Sciences and one of our newer board members at BioCrossroads. So Milan, great to have you. Excited for this conversation.
Absolutely. Thank you for having me, Vince.
Milan, why don't we just start off a little bit about a personal introduction and a little bit about your own personal journey and how you got to where you are now.
Yeah, absolutely. Well, I've been a proud Hoosier for a long time. I'm originally from Chicago, but I went to Purdue for my undergraduate and actually started doing cancer research as a freshman year in my undergrad, actually at the Purdue Center for Cancer Research over in the Hansen building. And started off, doing your typical slave labor as a undergrad. I was washing dishes, making Western blots, things like that. But eventually having my own projects. And at the time, you would not believe it, my project was on a marker called CTLA4 and CD28, which, as you may know, are some markers involved in revolutionizing immunotherapy. But we didn't know that at the time.
Super cool.
It is very cool. But anyway, from there, came to IU, got my PhD at the IU School of Medicine, and had the pleasure of being able to join the faculty right after my graduate work. And in about 2014, we started a clinical precision medicine program dedicated to using cutting-edge technologies for late-stage and refractory cancer patients. And we launched that at IU and IU Health. And it really grew from there, building a program, getting patients access to cutting-edge technologies. And then academically, rising, getting tenured, and had a really phenomenal tenure at IU in my research in precision medicine and precision oncology. And then in 2021, was recruited to Caris Life Sciences. I know maybe folks who are local may not be as familiar with this company, but Caris Life Sciences is based in Dallas, Texas. It's a large molecular information company that has really prided itself on providing as much comprehensive information to understand patients' cancers, to give patients the best shot at treatment for their disease. And so it's a company that had standardized on doing something called whole exome and whole transcriptome sequencing. So looking at all 20,000 genes in the genome at the DNA and RNA level, and then providing this to oncologists and patients across the country and in the world. Little known fact, it's actually the world's largest genome center.
Yeah.
Sequenced 200,000 patients last year alone.
Wow.
And we use that data to help our patient of today, but also a piece that I work on quite extensively is using the data to help the patient of tomorrow, whether that's drug target ID, new biomarkers, new signatures. And so there I've served as Chief Scientific Officer and also head of biopharma, and had the pleasure of also recently moving back to Indianapolis. I'd be close to family, and so and having a great time.
That's great. Thanks for the overview.
Mhm.
Certainly, I had pleasure working with Caris in my prior life when I was at Roche Ventana. So
Yes,
you guys are great customers of ours, of
still are.
of our primary staining solutions and immunohistochemistry systems. And so had the pleasure of touring some of your labs down out in the Phoenix area
Yeah.
several years ago. Um, let's maybe just walk through, I think there's so many amazing innovations happening in oncology, both on the diagnostics side and also on the therapy side. But let's focus our attention really on the patient journey on the diagnostic side. I mean, it really starts from a cancer perspective, really on the screening side. Obviously prevention is the
Hmm.
best case for people, is to find a precursor to cancer,
Hmm.
or at cancer at a very early stage, so that we can have best outcomes. What are some of the innovations happening from a screening perspective?
Yeah,
On
absolutely. Well, obviously, as you mentioned, the best way to bend the survival curve for this disease is to be able to catch cancer as early as stages. We know that when cancer is caught at its earliest stages, it's much more manageable from a surgery perspective, from a treatment perspective. And we know from survival curves, these patients obviously have a chance at cure and obviously living longer. Um, in my opinion, the technology that is going to revolutionize how we do screening is something called multi-cancer early detection, or what we call MCED. And what these are tests, in particular, they take a blood sample, use really sensitive sequencing techniques to identify the presence of a cancer signal. And as you may know from posts on LinkedIn and others, Karis is very bullish on this concept of using a multi-cancer early detection test. We've been personally excited by the data we've seen early on, achieving roughly 61% stage 1, 2 sensitivity with a test like this. And why this is important is when we think about screening, there's a lot of cancers that don't have a screening modality,
Yes.
right? We're all familiar with mammograms, we're all familiar with colonoscopies, skin exams, low dose CT for lung, and so on. But there's a lot of cancers. The reason why sometimes the survival rates are so poor is because they're caught so late. They don't become symptomatic until they're stage 3 or 4. So there is not a screening modality, a good one for things like liver cancer and pancreatic cancer and esophageal. So, what these tests really hold promise is to be holistic in our ability to screen patients. And in a particular area of strong interest is high risk individuals. And so, high risk individuals are folks like people who carry, for example, a germline mutation that predisposes them to cancer, or a strong family history, or folks who've had long term smoking or had exposures maybe in the workplace or whatnot. This is real opportunity. So, when we think about the future of MSET, I really believe that we're going to see a future where you're going to go into your doctor's office, you're going to get your, annual lipids, you're going to get your CBCs, and you're going to get a blood test to determine if you have cancer or not.
That's great. I love that. It's really the holy grail, really, isn't it?
It really is. It
And
really
even
is.
where you have modalities of existing modalities like colonoscopy, or other non-invasive ways to do colorectal screening, or and CT for lung, the compliance rates aren't great.
No.
And so to have a blood draw that could look at a number of different cancers, both that have existing screening modalities and those that don't, is really very compelling. So,
It
and
is.
what's promising about your data really is that early stage sensitivity, right? We've seen a lot of products out there that don't have great results with regards to early stage sensitivity. So to have you know sensitivities in 60% plus in early stage cancers is really where we need to be.
It really is. And it's been interesting because as we've seen this field evolve, it's become a real education battle out there because what you'll see out there is numbers quoted, and folks may not be familiar with they'll say it has this level of accuracy. Well, accuracy is a term that can be misconstrued. Sensitivity is saying, okay, if a patient has cancer, how often do we detect it? But there's also another piece that folks are not as familiar is this concept called specificity, which is meaning if you don't have cancer, the test says it's negative.
Yeah.
And what you can have is a test that has a lot of false positives, especially when you're screening hundreds of thousands of individuals. The burden to the health system for a test that has so much false positives is not feasible. And so, as I try when I educate, particularly patient advocates, I train them on this concept. Okay, look at sensitivity. Obviously, it's gotta be important, but also look at specificity because this is what's really, really important. We don't want to scare a bunch of people who don't have cancer. And then there is this concept, probably the most important metric, which is PPV,
Mm.
positive predictive value. So, out of all the patients test positive, how many in the end actually have cancer? And so, what we're seeing now, obviously, is wanting tests, and Caris obviously is at the tip of the spear of this that have maximal sensitivity, maximal specificity, and the highest PPV as possible to create the best tests to really try to bend the survival curve. Yeah.
So that's really the screening leg of the journey. And so if there's a positive signal,
Mhm.
you can have further tests, right? And so,
Yeah.
what are some innovations in that realm of initial diagnosis? Are we still quite reliant on the basic hematoxin and eosin that then triages to immunohistochemistry, or what's the state of the art there?
I love that you bring this up. It's interesting that many of the techniques that we use for diagnosis are things that have existed for over 100 years. Like, we we haven't. There's a lot of basic pathology that remains pretty mainstay. But one of the things that one of my favorite um, innovations at CARES is actually a tool we call GPS AI. It's a tool that we actually run in every patient we sequence. And what it does is it uses the DNA and RNA sequencing to determine the diagnosis, the tissue of origin from the sequencing data. And the reason why we developed this was originally for cancers of unknown primary. So this is a situation where a patient's diagnosed with cancer and the oncologist doesn't know where it came from. It's actually a really, really scary thing to
Yeah.
happen. Doc comes in the room, says, "You've got this awful looking cancer. We don't know what it is."
Hmm.
So you add more fear and uncertainty. And then the bigger issue is the fundamental basis of all cancer therapy begins with the diagnosis. It begins with what tumor, it determines what chemotherapy you get, what targeted therapy, so on and so forth. So we built this tool, trained on over 250,000 cases, a very powerful AI algorithm, and became fantastically accurate at determining tissue diagnosis. Now, where things got very interesting is when we launched this, we sequence about 700 roughly patients a day. And every day we get about 1 to 5 cases where the algorithm says it's a different diagnosis than what came out in the pathology report.
Mm.
These aren't people with cancer of unknown primary. They have a diagnosed cancer. But the algorithm is saying, you know what, this is actually looks different. And actually, our pathologists will take those cases, work up those cases with additional IHC stains and get to a better diagnosis. And so, what we have found is these tools have actually not only good for cups, but also good for getting patients to a better diagnosis. And
Right.
I actually think in the future, Vince, um, I would not be surprised if molecular profiling and digital path AI is what actually will be used for our final definitive diagnoses.
Yeah, no, I see that horizon as well, and which is exciting because I think what you're also underlying thesis behind everything you're saying is really what we've come to understand, that there is not one cancer, right? There's thousands of cancers,
There is.
types of cancer, and
There
and
is.
cancers moving away from being solid tumor specific, but really pathway or mutation specific, right?
It
Yeah.
really is. I used to tell patients. So the NCI, I think, has at last check 212 different subtypes, and but then when you break down those 212 subtypes at the molecular level, you're exactly right. It's almost an infinite number of combinations. And so as I used to tell patient advocates, you can take two patients with the same diagnosis, maybe the same age, race, grew up in the same town, maybe even have the same histology, but at the molecular level, each cancer has its own unique fingerprint. And that really is the basis of this concept of precision medicine that we've learned over a long period of time that if you take a group of patients with the same diagnosis and treat them all with the same drug, you don't get the same response. Some patients do well with a particular drug, some do not.
Mhm.
And so, this whole concept, as you alluded to, of using genes and pathways and genomic sequencing data is to really take advantage of the idea that each patient's tumor is unique and each unique tumor requires a unique approach to be able to attack it head on.
You are listening to BioSignals, the podcast powered by BioCrossroads. Let's get back to the conversation between Vince Wong and Milan Radovich.
So you've done the therapy selection using these really advanced tools
Mhm,
and gotten to a patient to a particular therapy regime.
mhm.
Um, w- what tools are now available to see, if those therapies have worked? What's emerging?
Yeah, so one of the greatest advances in cancer precision medicine in the last several years is this concept of the liquid biopsy. So, many of you are familiar with a tissue biopsy. A tissue biopsy is when a radiologist uses a needle to, take a piece of the tumor, whether that's from the liver or the breast or whatnot. A liquid biopsy is where we take a blood sample, use really sensitive sequencing techniques, and determine the molecular makeup of the cancer. And the reason we're able to do this is when we think about a tumor, we think about this ball of cells, for example, that's growing in a particular organ. But what's interesting, there's always a part of that tumor that is also dying either due to apoptosis or necrosis, primarily due to the lack of oxygen or nutrients. And when that happens, the DNA is shed into the circulation, and such that you could take that blood draw, extract the DNA from the plasma, and use these sensitive techniques to make up the molecular profile. Well, one of the cool things you could do with liquid biopsy is actually monitor either response or progression to therapy. What we can actually look at is, for example, is the increases and decreases in the levels of circulating tumor DNA. But also importantly, is that we can actually in real time monitor how the cancer is evolving in the face of therapy. And so, what we've seen over the years is studies of how cancers, when treated with a particular drug, will develop new mutations to be able to overcome exposure to a particular therapy, and then actually use kind of newer, cooler interventions. And so, what we've seen recently, is several pharma companies develop agents that go after what we call resistance alterations, going after these mutations that evolve in the face of therapy to be able to circumvent that.
It's really a cat and mouse game, isn't it?
It really
Yeah.
is. It really
Yeah.
is. To my friends in agriculture, they've actually dealt with something similar for decades, which is weed resistance.
Yeah.
The ability to, constantly having to deal with a new evolving strain or whatnot, and sort of a similar,
Yeah.
yeah,
Great
selection
metaphor.
pressure.
Yeah. Great analogy.
Yeah.
Um, and so how far away, I mean, is MRD, minimal residual disease monitoring is what you just described. Um, is that now considered a standard of care, and how widely, used is that, is MRD? And if it's not ubiquitous, what are the main barriers for MRD to be, used through in communities throughout the country?
Yeah, absolutely. My colleagues and I published some early papers in MRD years ago, actually, at IU. Actually, one of the highest cited papers in JAMA Oncology is a paper we did in MRD and triple negative breast cancer. And, this is an MRD kind of takes it another step earlier in the disease course where you've got a patient now who has completed therapy for curative intent. So they've had surgery, they've had chemotherapy, they have radiation. And what you do is you take a blood draw to see if cancer is present to determine whether the cancer is going to relapse. We know that a subset of these patients, the cancer comes back. We call this relapse or recurrence. And so, what we've seen through published studies is that MRD is a very strong predictor, maybe honestly the strongest predictor of cancer, of the risk of cancer coming back. And so, what we're seeing is a kind of new paradigm of clinical trials where we enrich patients who are at high risk of relapse and give them maybe an escalate therapy in those patients. But where I actually think the future of MRD is, Vince, where I think it will make the biggest impact is actually in treatment de-escalation. What we know from several studies is that there's a group of patients who are treated with adjuvant therapies who may not need it, who were effectively cured by their surgery, for example. We could potentially spare giving a ton of these therapies, which have toxicities,
Yeah.
or maybe it's potentially lessen the duration. And so I think the future will be the negative predictive value of these tests, their ability to actually say, you know what, you're okay. Will be, in my opinion, will be the future paradigm.
Yeah, and that's exciting. It really can potentially spare patients from the kind of
Yep.
pretty negative side effects of some that come with some therapies, right? So if you can preclude them from having to suffer through that, if they're not going to get any benefits, that'd be pretty amazing.
Exactly.
I know that Keros has a lot of collaborations with different pharma companies.
Mhm.
And so maybe talk about what that looks like, and what are the hallmarks of a successful collaboration between a diagnostic developer like Keros and a pharma company.
Well, as I always say at Caris, we pride ourselves on creating the best battle plans for patients, meaning providing a comprehensive genomic blueprint to able to identify alterations and guide therapy. However, the best battle plans also need the best weapons against the disease. And working with cutting edge pharma and cutting edge biotechs to be able to take that information and create the next, game changing therapy is really quite important. It's quite important to our ethos. Precision medicine in itself is predicated on the concept of having medicines that are precise and being able to go after these particular genomic alterations. I will tell you, in my role as head of biopharma for CARES, just the sheer amount of cool Star Trek level of innovation I'm seeing out there right now is mind-boggling. Things like personalized vaccine therapies, things like in vivo CAR T against solid tumors, novel ADCs with the most coolest payloads that have neat, cool biology. I mean, what we're seeing out there right now with some of these newer agents is response rates we never imagined 10 years ago. never imagined 10 years ago. And so one of the things is not only is it cutting edge, but as these agents now move into the curative setting, I
Yeah,
think we're going to continue to see obviously considerable improvement in survival with these approaches.
that's very exciting.
Yeah.
Um, how are how is Caris and maybe diagnostic companies at large leveraging AI
No.
in terms of developing products, whether it be you know uh, algorithm development or data analysis or regulatory submissions or clinical trial enrollment? Maybe
Yeah.
maybe talk a
Yeah.
couple of use cases of how
Yeah.
you're utilizing and leveraging AI.
I'll try to keep it brief because it would take too long. But AI has been in our ethos long before it was a buzz term and long before ChatGPT wrote your kids' essays. I mean, this is something that has been something we don't, we used to call it machine learning,
Machine learning,
as you're well aware. And now AI is the ubiquitous term. And so we have a very large group of these super bright PhD mathematicians and computer scientists who all day really work on developing the next algorithm and approach. Um, why AI is important. So what we let me talk very high level of what's made it possible. It has been the convergence of high throughput molecular sequencing plus
big data sets.
big data sets. You've had to have big data sets plus novel algorithms and mathematics in combination with the advent and explosion of cloud computing. If you didn't have those three converge, we would not have what we have today. It required really all three to make it happen. And so you're exactly right. For Caris, to our knowledge, we have the largest data set in cancer. We've sequenced over 1 million patients, over 700,000 whole exome transcriptomes. And the AI is only good as you train it. So you need these massive data sets. And so we are constantly using these data sets to do things, for example, develop a next biomarker for therapy prediction, or like I said, for more efficient diagnosis, or predict which patients could recur. One of the things that use cases that I'm super excited about is this concept of digital path AI. It's taking a H&E stain that has existed for decades, and the AI algorithm, without any sequencing, can look at that H&E and say, you know what, this is the mutational makeup of the cancer.
Yeah,
It's unbelievable.
fascinating. Yeah.
Predict what therapies it could respond to. Predict its recurrence. We just recently launched a tool called My Clarity that actually predicts the up to 15 years recurrence risk of an ER positive breast cancer just from an H&E. Well, when you think about this, Vince, think about the implications, right? One is the turnaround time. You're
Yeah.
talking about something that could be turned around in maybe in theoretically minutes, but it also democratizes it, right? It enables technologies that were limited to the US or academic medical centers, and now you can make it available around the world
Excellent.
because you upload an image to a server and let it get an answer back. So to me, I'm very bullish on digital path AI. I really think as we look into 26 and 27, it's going to revolutionize what we think this area of precision medicine.
Yeah, when I was working in pathology 10, 12 years ago, right, we were trying to push a string up the hill of getting pathologists to adopt digital pathology. And I think, now I think we're beginning to hit that proverbial tipping point as a lot of different factors kind of converge to really enable the adoption and embrace. And I think, you're seeing some strategic acquisitions from some key players like Roche and the
You are.
acquisition of Path AI that really is I think, a hallmark of what you're talking about, right?
Yeah, you really, really are. Now, to my pathologist friends out there, I get this question often. No, I don't think you're being replaced. Okay. Well, I think pathologists, but the way we're seeing this is, sort of this lab in the loop concept. It's these tools making pathologists better at what they do. It's these tools making radiologists better at what they do. Anything that involves pattern recognition. This is where these AI tools are becoming really, really strong.
Yeah,
Yeah.
super exciting. Um, Talk about Indiana and the ecosystem here, right? You've been trained here and you've moved back and then you have purview and visibility to ecosystems throughout the country and
I
even the
do.
world. Maybe narrate for me a little bit about kind of observations of the dimensions that you see and characteristics you see here in the Indiana ecosystem.
You know, um, I'm very, very bullish on this ecosystem, as you're well aware. I see something very, very special growing. Um, I know many of us who've lived here in Indiana have had to deal with many years of being, saying, well, flyover country, a small town, what could it be? Yet, when you look at the productivity, we've got the most valuable pharmaceutical company in the world. We're the radioligand pharmaceutical capital of the world. We got massive investments in diagnostics, as you're well aware. Orthopedic capital of the world.
Right.
I mean, it goes on and on. Premier universities, massive health systems. And so when I look at Indiana, I look at an ecosystem that is literally going exponential right now. And for the longest time, people would look at this concept of, epicenters of biotechnology or whatnot, and they would think about South San Francisco, and they would think about Kendall Square in Boston, and so on and so forth. But what we have here is super special. What are some key differentiators, though? I think one is we can attract talent, especially kids coming out of our major universities, offer a relatively lower cost of living, and be able to do some really, really cool science at many of these institutions that we have here. We also have, I think, an ultra collaborative, I don't mean to overemphasize it, but we're Hoosier hospitality. One thing I've noticed about Indiana is that when you go talk to another scientist or another company, everyone is willing to collaborate, work together, shake hands, innovate together, figure out how to way to do it. And so, when I see what's happening here, I'm not surprised just by the level of growth that we're seeing.
Yeah. Innovation is a team sport, right? And so you have a mindset of collaboration here.
It really is, and now we've got the best college football team in the country too. And I'm thinking to myself like, for all these years, but yeah, Indiana's is kicking rear end, and taking names right now.
Yeah.
And so I'm very proud of this, very proud of this state.
Yeah. And you've had some interaction with the Indiana Bioscience Research Institute as well, I think, right?
100%,
With Alan
yeah.
and his team. Yeah.
With Alan and his team, it's fantastic. Really working to develop a next generation of therapeutics across a variety of diseases. And I think as we look in the future, it's really and Vince, I know you're at the forefront of this, but it's creating an ecosystem of being able to create the next generation of companies who both have really cool science, who have access to capital markets, who have access to resources like we have at IBRI and others to be able to advance the next generation of therapies.
Yeah, that's great. Well, we're lucky to have you here in the bio heartland where science solutions are invented, made, and moved. And you're, just such a bright, capable, and accomplished leader that we're just lucky to have in our ecosystem. So, Milan, really enjoyed the conversation. Look forward to continue to partner with you to grow this ecosystem and also help enable your success too. So, thanks for taking the time.
Great. Thank you so much for having me, Vince.
For more information about BioCrossroads, our events, programs, and how you can get involved, please visit biocrossroads.com.